Good afternoon, everyone, and thanks so much for joining us for our Leaders Initiative seminar series. Our speaker today is Dr. Jessica Damoiseaux. She is an associate professor in the Institute of Gerontology in the Department of Psychology at Wayne State University. Her research program aims to understand the changes in brain function and cognition that accompany normal and abnormal aging. So with that, welcome, Dr. Damoiseaux.
Thank you, Courtney, for the introduction, and also thank you for having me share some of our research here to this group of people. So some of the work that we're doing in my lab is to look at brain aging and sort of looking at what is typical and when do we see that it might be pathological. And what we're really interested in is sort of here, what you see here, that difference between normal aging, and here, the trajectory of going towards mild cognitive impairment and dementia. So in typical aging, you would see that sort of everyone has some slight cognitive changes. So you see some decline in cognitive function and also in brain structure and function. But in here, for those individuals who are on a trajectory for dementia, you see that at some point there is a more steady and then a more fast decline in their cognitive abilities and in their brain and function and structure. So what I'm interested in our lab is specifically looking at this stage here. So those individuals who may be on that trajectory, but at this stage, they are still, they don't have any symptoms yet, so they still perform cognitively relatively normal, but they may be on this trajectory and not on sort of that normal aging. So some ways that we're investigating these groups and sort of see if we can detect any early atypical changes. So one line of our research is into subjective cognitive decline. So subjective cognitive decline is that older adults who have complaints, memory complaints, and they're worried about those memory changes. But when they go to the doctor, they get tested for some cognitive performance, and they still perform within the normal range. So the question here is like, are these individuals, is it normal aging? So can they be sent home and just say like, hey, this is just typical aging, don't worry. Or might they actually be on this trajectory here in the light blue that you see here? Could it be that this is the stage where the patient actually knows that something's going on, but the doctor doesn't? So that's something that we're interested in examining. Then another line of research that I wanted to present here is some of our work that we're doing in blood markers of neurodegeneration. And the idea here is that can we detect some early changes or atypical changes by doing a blood draw and look at this marker of neurodegeneration, which in our case is neurofilament light, and can that already indicate that maybe some individuals may be on this trajectory of pathology and not typical aging. So I'm going to start to talk about subjective cognitive decline and our research into that. I'll focus a lot on functional connectivity. That is one of the main approaches to examine brain function that we use in my lab. But I'll also include some of our research where we also looked at hippocampal volume, and we also looked at white matter hyperintensity burden and vascular risk associated with that. And then I will switch over and talk a little bit more about our research on blood neurofilament light and the association with hippocampal volume, long-term memory, and functional connectivity. So let's start with the research on subjective cognitive decline. And as I already just mentioned, so subjective cognitive decline, it's these older adults who have memory concerns but perform normally on cognitive tests. So they are just typical older adults in that sense. However, some research has shown that, and you can see that here, that those older adults who report these complaints, that they are more likely to develop MCI or Alzheimer's disease than older adults who do not mention do not have those complaints. And then some early work looking into this has shown that these older adults who have subjective complaints that they, in this case here, have lower left hippocampal volume than older adults who do not have these complaints. So, as you may know, is that hippocampal volume does tend to decline with older age, but what you would see here is that it's more of a decline than what you would typically see in age in these individuals. And hippocampal volume also is known to decline in Alzheimer's disease. So it might be that these individuals here show some hippocampal volume changes that could be already indicative of some Alzheimer's disease. So what are the typical changes that we see in Alzheimer's disease? I know for most of the people here, you're probably familiar with this, but I'll go over it anyway. So what I already mentioned here is the atrophy. So you'd have the hippocampal volume decline, but you would see that atrophy sort of all throughout this network. So in the medial temporal lobe, but also here posterior cingulate precuneus regions and then frontal regions. So all initially, sort of earlier on in the disease, you would start seeing atrophy throughout this network before it spreads more throughout the brain. And what Randy Buckner showed here in when he presented this figure is that this atrophy pattern really overlaps with glucose metabolism disruption that you see in Alzheimer's disease, which also targets these same brain regions, the posterior cingulate precuneus area and the temporal lobe and some frontal regions. And this also overlaps with amyloid deposition, as here I think it's a PET imaging. And this sort of specific structure and network of changes that you see here actually overlaps also with this network, so posterior memory network that also includes hippocampus, posterior cingulate precuneus, and then some frontal lobe as well. And so that makes sense, of course, because also the first symptoms that you would see in Alzheimer's are also related to memory function. So you see this overlap in this pattern of brain changes related to memory network. Then what he also showed here is that this network, this pattern that you see across these different measures also overlaps with this brain network as measured with resting state functional connectivity. And this is what we call the default network. So this is a set of brain regions that is more active when we're sort of have intrinsic processing. So things like mind wandering, self-referential processing. When we're doing those things, you see activity strongly in this network. It is deactivated or less active when we're focusing on external stimuli. So this default network has been associated sort of with memory function and also with Alzheimer's disease. And as you can see here in one of the first studies that looked at this by Michael Gracious, but it's been replicated multiple times after, that specifically this default network connectivity that you would see lowered in Alzheimer's patients compared to older adults. So sort of trying to link this now to the subjective cognitive decline. So if we think subjective cognitive decline may be really an early stage, or these individuals may be on the trajectory for Alzheimer's disease, what would be sort of the regions where we expect the earliest changes? So this model here shows, again, this default mode network, and shows that especially this posterior part of the default mode network, so including the posterior cingulate and precuneus, that this is highly active throughout our life. So this is a central hub in the brain and is very active. And because of that, it also makes it more vulnerable with older age and potentially also genetic risks such as APOE4. So you would see that this, at some point in older age, this may start declining, but then if it's associated with the pathology, it actually may spread pathology, so noisy signal and some amyloid plaques, and maybe also the tauopathy may spread throughout this network. So the idea is that mainly this network is initially affected, it's spreading throughout the network in Alzheimer's disease, and that the initial part where you would see it is sort of in this posterior region. So this is the network that we focused on for our analysis that we've done to look at functional connectivity in individuals with subjective cognitive decline. So, and this was a research study done by Ray Viviano. He was a graduate student in my lab. And while in my lab, he examined this in 83 healthy older adults. And he particularly looked at this posterior default network or posterior memory network and selected these specific nodes and examined connectivity within this network. He also examined connectivity in the anterior memory network, where he didn't find any effects. So, but in this posterior memory network, we found that individuals who reported subjective cognitive decline and who were worried about it, that they had lower connectivity than older adults who did not report any complaints. And as I just mentioned, so this posterior network, so this is really where, according to this model for Alzheimer's disease, where we would also expect to find early Alzheimer's related changes. When we examined it more closely to see which specific part of this network seems to drive this effect, and we found that it was really this retrosplenial and then posterior singular precuneus. So really here in that area where we see the largest effect of subjective cognitive decline. So this seems really supporting that subjective cognitive decline, that there actually may be something more going on, and it's may not be just typical aging. And we know that this default network is affected. However, if you compare our results to some of the other studies that looked into this, there are some studies that show similar findings, but also some studies who actually show the opposite, that there was higher functional connectivity. So when trying to make sense of this, Ray looked into this, and he actually suggested that maybe when we look at these individuals, that it may depend on like where are they in the trajectory of changes, that we may see different effects. So maybe earlier on, we actually see some increase in functional connectivity that could be sort of to those noisy signals, increased signaling, and potentially also for compensatory mechanisms. But then later on, you see that it's really just through neurodegeneration, you see that it decreases. So to look into this a little bit more, we did a longitudinal study to examine this. So in this longitudinal study, he examined whether subjective cognitive decline would predict functional connectivity change between an examined hippocampus to the rest of the brain. And we expected to mainly find an effect within these regions of the default network, as these are again aligned with our cross-sectional study and the earliest regions that we would expect any changes. So we expect a decreased coupling between posterior cingulate and the fold regions. And he examined this in this whole healthy older adult sample of 69 older adults at the first time point, and then with 34 and 28 on the second and third occasion. Overall, it was about 3 years with about 19 months between measurements. Individuals were about 68 years of age, mostly female and mostly Black Americans. So Ray used quite a sophisticated approach to analyze this. So he used the whole brain voxelized analysis approach with using latent growth models as implemented in Neuropointillist. So I'll just briefly take you through this. So what he did is for each individual, so we have, for most individuals, we have 3 time points, and he calculated functional connectivity from the hippocampus through the rest of the brain. So you get these functional connectivity maps for each time point. So and these are put in the model as the first time point functional connectivity, second time point, and third time point. And then we examined the intercept and the slope, so they change over time in functional connectivity. And then assessed here the MFQ. This is a measure that we use to assess subjective cognitive decline. So this is the memory frequency questionnaire, functioning questionnaire. And he examined this. So this is a continuous variable. So opposed to the cross-sectional work where he examined two groups, people with subjective cognitive decline, those without. Here, it's included as a continuous variable. And then we also included age in the models here. So then from on the voxel, this was done on the voxel level. So then for the voxelized maps, there's been some thresholding and some fit indices to come up. And then here, with then also correcting for multiple comparisons, to come up with a final map of significant results. So what he found in this study, so this is all from the hippocampus. So from the left posterior hippocampus, we found that there was a region in the dorsomedial prefrontal cortex here where he found a significant effect, where subjective cognitive decline predicted a decrease in functional connectivity. And you can see that here. So this is again our measure for subjective cognitive decline. And you see here that that predicts a decrease in the change in functional connectivity over our 3 time points, so over about 3 years. For this specific model, we also found an effect here on the intercepts, on just the level of functional connectivity, sort of indicating, and you kind of see that here, this is, for illustration purposes, is divided into low and high subjective cognitive decline. But again, it was used as a continuous variable. But it indicates that those with initially higher subjective cognitive decline was associated with higher functional connectivity, but then a steeper decline in functional connectivity over time. So this actually could fit well with that idea that Ray had of that maybe initially you see an increase in functional connectivity, but then over time you see that it actually decreases more, so you get this steeper decrease in functional connectivity over time. Then for the right posterior hippocampus, we find a similar effect, so also in the dorsomedial prefrontal cortex. And again, this is a voxelized analysis, so it's really interesting to see that for both left and right posterior hippocampus, we do find sort of that dorsomedial prefrontal cortex that's associated with this. And we find a similar effect here, so that subjective cognitive decline predicts decreasing functional connectivity. So we did not find an effect here on the intercept, but we did see the same effect on the decrease over time. So, and then the posterior hippocampus and the connection with the medial prefrontal cortex. The signaling between these regions does seem to be very important for long-term memory functioning and also some social cognitive processing. So that fits really well with the complaints that these individuals have, and also potentially with some of the deficits that you could see in Alzheimer's dementia. So we were also interested in, do we actually see that subjective cognitive decline may also predict a change in cognitive function? So per definition, all these people with subjective cognitive decline, when they get sort of that label as subjective cognitive decline, that's because they do not have any objective cognitive deficits. But we thought that maybe we will see some change in cognitive function over time. So we examined here working memory and delayed memory, but we did not find any effects of subjective cognitive decline. So there was subjective cognitive decline was not associated with overall cognitive functioning and also not with the decline over time. We did find age effects, which you would expect. This has been very consistently found in the literature that age with older age have lower functioning and for memory functioning. So we see that here for working memory, and then for delayed memory, we see that it's a lower performance and also a larger a decrease in performance over the 3-year period. So then, in addition to looking at those functional connectivity effects, we were also interested in examining the association with hippocampal volume, as hippocampal volume is sort of still the gold standard in measuring neurodegeneration in Alzheimer's disease. So what we were interested in here was to examine if we could see that in a trajectory of changes, does sort of functional connectivity, does that predict maybe some changes in hippocampal volume? Or could it be the other way around, that hippocampal volume predicts changes in functional connectivity? Sort of what is that lead lag effect? And so we examined this, and let's see. So what we found is that here, so again, the measure of subjective cognitive decline, that in this model here, we see that it does predict the change in gray matter volume over time, so that there is a larger decline in hippocampal volume in those with more subjective cognitive complaints. However, we do not see any significant effect on functional connectivity in this model, and no interaction with gray matter volume and functional connectivity. We do see that for age. So we do see that older age is associated with lower functional connectivity over time. And we also see that gray matter volume mediates that association, so that in those individuals, and I think it's actually those who have lower hippocampal volume, then you see a larger decline in functional connectivity. And that was for the left hippocampus, and we find a similar effect for the right hippocampus. The only thing is that this direct age and functional connectivity association is not significant here for the right hippocampus. But otherwise, we find the same effects of subjective cognitive decline on predicting a larger change in hippocampal volume, and that age, sort of indirectly here, predicts change in functional connectivity. So the conclusions for a subjective cognitive decline research in using functional connectivity is that we do see lower default network connectivity in individuals with subjective cognitive decline. And they also have a larger decline in default network connectivity over 3 years, and a larger decline in hippocampal volume over that same time period. And then in aging, we do see a lead lag effect of hippocampal volume loss, and then decrease in default network connectivity. So then switching a little bit from functional connectivity and looking at some other brain measures that we want to associate with subjective cognitive decline. And this is work that Yujin Zheng, a graduate student in my lab, performed, where she was interested actually in white matter hyperintensity burden and how that's associated with subjective cognitive decline, and also the vascular risk factors, how they are associated with that. So this is a cross-sectional study where she first examined white matter hyperintensity burden using LST toolbox. So you can see that here. So for the whole brain, you can measure white matter hyperintensity volume. And then in addition to looking at whole brain, she was also interested in looking at specific white matter tracts. So she masked the whole brain measure with these white matter tracts. Here, the superior longitudinal fasciculus, here the forceps minor, and here the anterior thalamic radiation. So, and then what she examined was to see if a mean arterial pressure, so that was our measure of vascular risk. So does mean arterial pressure, does that predict, or is that associated with subjective cognitive decline? So, and this is the frequency of forgetting. This is actually a subscale of the memory functioning questionnaire that I mentioned before. So this is the same measure that we've used before for subjective cognitive decline, a continuous measure. And what she found is that there was no direct effect, but that whole brain white matter intensity burden mediated this association. So that individuals who have higher mean arterial pressure, that predicts they're more likely to then also have more white matter hyperintensity burden, and then which would then predict more subjective cognitive decline. And she found this effect over the whole brain, and also specifically for the anterior thalamic radiation. So for this track that you see here. but did not find that effect for the other two tracts, so not for the forceps minor and the superior longitudinal fasciculus. Then, in addition to looking at sort of this mediation of white matter hyperintensity burden in the effect of vascular risk on subjective cognitive decline, Eugen was also interested in looking at how does genetic risk, so here if you're an E4, ApoE4 carrier, how does that affect this association? And she actually found here that there was a moderation of ApoE4 carriership on the association between mean arterial pressure and whole brain white matter hyperintensity volume, so that individuals who have higher blood pressure, that it was specifically then those who are also E4 carriers that would predict having more white matter hyperintensity burden, which would then lead to subjective cognitive decline. So that this association is particularly relevant for individuals who are E4 carriers. This association, the moderation effect was not on the link between whole brain white matter hyperintensity volume and subjective cognitive decline. So it's really this association between blood pressure and then getting white matter hyperintensity, hyperintensities, that is really more associated with those who are E4 carriers. So what we showed in that study was that larger white matter hyperintensity burden is associated with more subjective cognitive decline. It mediates the association between mean arterial pressure, so risk factor, vascular risk factor, and subjective cognitive decline, and that it's particularly ApoE4 carriers with high blood pressure that have high white matter hyperintensity burden, which would then lead to the subjective cognitive decline. So that was what I wanted to share about the subjective cognitive decline. So now I'm going to switch and talk a little bit about our work looking at blood NFL as a neurodegenerative marker for Alzheimer's disease. And so to give a little bit of a background on neurofilament light. So neurofilament light is a sort of a proxy measure of axonal damage. So it is here part of the cytoskeleton that provides structure to our cells and our axons here. So if there's axonal damage, the neurofilament here breaks down, and some of these fragments get into the cerebral spinal fluid and into our blood. And this is typically, before it was measured in cerebrospinal fluid, and now there's some newer technologies that we can also measure this in the blood. So of course, the benefit of measuring this in the blood is that it could be with just a relatively simple blood draw that older adults could do at the doctor, that it has a potential to then indicate if this individual may already show neurodegeneration. And if this should be an individual that, if they have, for example, cognitive complaints, subjective cognitive complaints, that it could be an indication if they also have in their blood draw, if they have a neurodegeneration marker, that could be an indication to really follow up on these individuals and have them sent for more thorough examination. So previous research has already shown that neurofilament light has a potential as a neurodegenerative marker for Alzheimer's disease. It is not specific to Alzheimer's disease, though, because it is just a general marker of neurodegeneration here, of the axons. But it could also be an indicator for Alzheimer's disease. And it has shown that it can predict cortical atrophy, glucose hypermetabolism, cognitive function in individuals on the Alzheimer's continuum. So one of the studies here that shows this is in cognitively unimpaired older adults who are amyloid positive. And here you would see that, that there was an association with higher plasma neurofilament light that was associated with here, so what is it, lower metabolism, and then here in the same participants that you would see that it was also associated with a lower gray matter volume. So higher plasma NFL, lower gray matter volume. And if you look at the specific areas the brain that are affected across these studies. And typically you see, you can see that in these examples as well. So you see the temporal lobe, including hippocampus, but also again, these posterior default mode regions that consistently come up as regions that show an association with neurofilament light. So with Eugen, and this is Eugen's dissertation work that she's doing. So what she planned to do is to replicate some of the previous findings between on the association between plasma NFL, hippocampal volume, and long-term memory, but then also examine the association of plasma NFL with the fault network connectivity in cognitively impaired and unimpaired older adults. And I'll show you some of the results, preliminary results that we have from this work on hippocampal volume, long-term memory, and functional connectivity. So in here you see the sample. So we have 47 cognitively impaired individuals. So these are individuals with amnestic MCI and Alzheimer's disease. And these participants are actually from the MADC UNMEP dataset. So this is a collaboration with Ben Hemstead and with Scott Counts and with Drew Bender. So we're really working across University of Michigan, Michigan State, and Wayne State on this project here. And some of our preliminary findings show that there's no significant difference here in the level of neurofilament light that we measured between cognitively impaired and unimpaired older adults. But we do see that association that has been found in the literature. So we replicate the finding here, and this is with a measure of delayed, so the long-term memory, but it's actually, so it's delayed recall, and it's a factor score based on multiple measures of delayed recall that we have. And here you see that specifically in the cognitively impaired individuals, so those with amnestic MCI and Alzheimer's disease, that you see that those who have higher NFL measure, that they have a lower performance on this task. We do not see an association in the cognitively unimpaired group here. And so this is in line, this replicates previous findings, so that's reassuring to see. And then we also looked at hippocampal volume, and we find a similar effect, so that higher neurofilament light in the plasma is associated with a lower hippocampal volume here in the right hemisphere, and we do not see that association in the cognitively unimpaired older adults. And we found a similar effect here for the left hippocampus. That is also cognitively impaired. We see an association, not in the cognitively unimpaired group. So then for sort of the newer part of the work, looking at the association between plasma NFL and the fault mode network functional connectivity. And I'm just showing you all three of our measures at once because we found similar effects. But I'll describe them briefly. So we had measures of default network functional connectivity. We have three different ones. So one is really just the functional connectivity within that posterior singular precuneus node. And then we have another measure, functional connectivity within the default network of this posterior singular precuneus node to lateral parietal regions. And then another one here, posterior singular precuneus node to medial prefrontal cortical regions of the DMN. So it's all within the default network connectivity. It just sort of divided into more posterior to anterior, medial to posterior, medial to lateral, and really just specifically in that posterior hub. But as you can probably see here from these graphs, is that overall we did not find an association between neurofilament light and functional connectivity. And we also did not find any differences across the groups. So across the cognitively impaired and unimpaired, we did not find an association between functional connectivity and these measures. So that was a bit disappointing, as we had hoped that functional connectivity might have been sort of an early indicator that would link with the observation of neurofilament in the plasma, but it did not. So we did find, however, so consistent with previous findings, we did find higher plasma NFL was associated with more hippocampal atrophy and lower long-term memory function, but our results do not support the association between plasma NFL and the full network functional connectivity. So what's next is that we were also going to analyze diffusion imaging measures to look at the white matter integrity. So as I mentioned, is that neurofilament light is specifically a measure of neurodegeneration of axonal damage. So we think that white matter integrity is actually looking at these connections, these structural connections, may possibly be an earlier indicator of change. So we're going to use this approach, neurite orientation, dispersion, and density imaging models, where we can look at different measures of the structural connectivity. So we can look at the neurite density, we can look at free water, and orientation dispersion as well. So this is what Eugen is still working on currently, and we're hoping to get that done in the next couple of months. So then to wrap up, the idea, so does this research, does it give us an idea about typical versus pathological brain aging? So the research that we've done on subjective cognitive decline, we can confidently say that it does sort of give evidence or supports the idea that individuals who report this, that they actually may be on a trajectory, sort of more this blue line, so that they do show some evidence that from their brain measures that they actually may even end up being on this trajectory. Of course, it's a heterogeneous group, so there may also be individuals that end up just staying sort of right there and not declining as rapidly into MCI and dementia. But we do have indication that they are overall not on sort of this normal aging trajectory. However, for our blood marker of neurodegeneration, the results are somewhat not as conclusive as we'd hoped. So what we can see, does it really detect early changes? At the moment, most of the research actually supports more that it may not really be a good indicator for really early changes. So at this stage, as you can see here, as a monitoring biomarker, that you see this prediction does not seem to be holding up. It does seem to be associated really with individuals who already have mild cognitive impairment or maybe Alzheimer's disease diagnosis. So as a monitoring biomarker, it seems to be associated more with those individuals who already have other symptoms as well. And then also for the studies so far sort of show that it might also be good as a prognostic biomarker, so that individuals who already have disease symptoms or pathology, that it can predict also then their change over time, their prognosis. The thing that we still have to look at, and at least our research does not support sort of that the measure at sort of a moment that individuals are still within the normal range, so healthy older adults without any other symptoms. It does, at this point, whether or not it predicts sort of whether they develop the pathology, that's not clear yet. Definitely not with our functional connectivity. And even our measures of gray matter volume seem to be associated more with individuals who already have disease symptoms. And so this may be something that with a diffusion, hopefully we might see some of this potential here for risk susceptibility. So that's still work in progress. So that's what I wanted to share with you. So I do want to acknowledge my lab members and previous lab members and collaborators on these projects and my funding. And please let me know if you have any questions.
Thanks so much, Jessica. So everyone can just unmute if you have questions whenever you're ready. Just jump on in.
I have a question. Yes, it's Amanda, but I see a hand raised, so maybe that person, feel free to go.
Just whoever is ready to go first, just go in. Amanda, you can start.
Okay, so hello, Jessica. Good to see
Hey,
you.
Amanda.
I guess one thing I have heard, and I could be totally wrong on this, is that in like early to mid stages of dementia, individuals may be less aware of their memory impairment or cognitive decline. So I guess one question I have is, do you see any timeline for when these reports may be more or less accurate in terms of their association with what you're seeing neurologically?
Yeah. Yes,
Does that make sense?
yeah, no, it does make sense.
Yeah.
And yeah, it's a good question. So yeah, there's definitely, I think you have some individuals who sort of early on notice it, other individuals who don't notice it, notice their own change, are not aware of their own cognitive impairments. We have not specifically looked at that. We did look with a longitudinal study over the 3 years. We did not see any change in their reporting of their memory complaints and concerns. So that's the only thing that I can say, that we looked at that and to see, like, do we see that their complaints change over time? And we did not see that change. And I'm actually not aware of many studies that have really tried to track that and see, like in individuals, so how does it over time? Do we see variation in how people report their complaints, and how does that link to actual pathology or cognitive, objective cognitive decline? So that's a really interesting question, and I don't have a good answer to that.
That's okay. No, thank you.
Hi, I have a question for your longitudinal study in the beginning. When you do the seat-based connectivity from the hippocampus, do you take like the whole hippocampus, or do you just take a part of it? Or like depending on the resolution, of course.
Mm-hmm.
But from our studies, we see it's more like the default mode network projects just to parts of the hippocampus. So if you take another seed region, you get a better projection of your default mode network. Would that have any implications on those results?
Yeah, so we looked at the posterior hippocampus. And yeah, I've done, I didn't show that here, but we've done previous work also just in typical aging, that we looked at the difference between anterior and posterior hippocampal connectivity patterns, and that we do see that, yeah, posterior hippocampus is really more linked and connected to this posterior DMN system. And actually, I think, yeah, let me I like go back quickly, because I can show you for the cross-sectional study that we did, but it's is this similar region. Oh, so I have to go back quite a bit. Where we looked at here. So here you can actually see, and it's not very clear because this is just an illustration how we posted it here. But here we actually looked at the difference between that anterior and posterior memory system, whereas the posterior memory system has the posterior hippocampus to, as you can see, all these regions here, and the anterior memory system has anterior hippocampus to more these sort of temporal polar, and then here frontal regions. So it's really based on that, the difference between that anterior versus posterior hippocampal connectivity patterns. All
right, great, thank you.
Yep, you're welcome. Yeah, so I see in the chat, sort of looking at it, if we've looked at other plasma biomarkers. And we haven't done that yet. So we're sort of starting with NFL, but the goal is to see if we can also get some other markers and see maybe some other markers are more sensitive as well. So that's something that we hope to also collaborate on more, within the MAD-RC with the biomarker core, that we can look at other blood markers as well. So this was really just our first foray into linking blood biomarkers to neuroimaging, but we're hoping to continue that work. Any other questions? I'm just going to go back to my last slide because it also has my email address on it. So if anyone, maybe later on, if you think of anything, feel free to contact me. I'm happy to answer any questions later on as well.
Great. Thank you so much, Jessica. This has been very interesting. Looks like we have a couple more hands raised for questions. So whoever wants to go first, just go ahead and unmute yourselves. Oh, those weren't hands raised. Okay.